Good morning, students. So we are discussing regarding the cytokinesis. So karyokinesis we completed, and in cytokinesis, as I discussed in the last class, it is of the division of cytoplasm. We call it as cytokinesis. In animal cells, it is cell-furrow method. In animal cells, the division is called as cell-furrow method, whereas in plant cells, it is a cell plate method. It is called cell plate method. Okay. So what is this? And what is this cell-furrow method? And in plant cells, let us discuss. Okay. So keep the heading cell-furrow method in animal cells. So here, if you see in the cell-furrow method, a shallow groove. A shallow groove so this shallow groove, or you can call it as a constriction or constriction. So the cell itself will constrict into two halves. A shallow groove or furrow in plasma membrane formed in the furrow formed in the plasma membrane. Okay, so this, whatever it is formed, that will deepens to join. So the furrow deepens and joins in the center which divides cytoplasm Okay, that means it is of okay, right. So whatever the cell, initially you see the formation of two nucleus. So this will deepens finally to divide into two cells. So it constricts to divide into two cells. So such kind of the method, we call it as cell-furrow method. A furrow is formed. So this is called furrow or deepens. Okay, and divides the cytoplasm into two different cells. So this method is called cell-furrow method, which is especially in the animals. Now, if you take the plant cells, so let us see what happens in the plant cells. How does they divide? As you already know, in the animal cells, there is no cell wall. That is why the cell membrane will deepens easily to the center. So furrow can be easily formed. But if you take in the plant cells, it has cell wall. A thick and rigid cell wall is present. So it cannot go for furrow method or constriction. So it has to, the new wall develops from the inside to periphery outside. So we see the formation of a plate-like structure from the center. Okay, so that is why we call this as a cell plate method. So second one, the plant cells. Here, the remnants of spindle, how does the plate is formed? How does the cell plate is formed means the remaining fibers, see when the spindle fibers break, then automatically you see the remains, those fiber like structures which are present in that particular cytoplasm, those are the remnants of the spindle fibers. Those remnants of the remnants, remaining parts, REMNANTS, the remnants of spindle fibers. The remnants of spindle fibers forms barrel shaped phragmoplast. Shape phragmoplast. So this is it forms a new structure called as a phragmoplast with the spindle fibers. To this phragmoplast, calcium, so it adds to this calcium and magnesium is added. So it adds the calcium and magnesium by Golgi complex. By the Golgi complex, the calcium and magnesium is added and forms into a liquid cell plate. And forms a liquid cell plate. So this is not solid, liquid cell plate, it is formed. That is called precursor of middle lamella, which is a precursor of middle lamella. So this is the starting of middle lamella. With the precursor of middle lamella. Okay? So middle lamellum is formed initially from this cell plate only. We tell middle lamella in the plant cells. So this is formed the formation of this middle lamella is this origin. First phragmoplast, that means the spindle fibers, calcium and magnesium are added, and that will form the liquid cell plate, which is a precursor for the middle lamella. Okay, right. Then that will grow centrifugally. This grows centrifugally. That means centrifugally means something from the center to away. Away from the center is called centrifugally, and joins with parental cell walls. This will join with the parental cell wall on both sides. On both sides. That means whatever it is formed, whatever the cell plate which is formed, that cell plate which is, see, initially, say this is a cell wall, okay, cell wall. So in the plant cell, at the center, from the center the movement will take place in this direction. So this is centrifugal from center to away and forms a barrel shaped structure, that is called as phragmoplast. To this calcium and magnesium are added, spindle fibers, initially it is called liquid cell plate, and that cell plate will touches the cell wall on both the sides, and it is forming the division. So this is called middle lamella. So now it divides into two different. Later, gradually, you can see, gradually it solidifies. Gradually it solidifies and transforms into solid middle lamella. You can write that point. Gradually the cell plate, the liquid cell plate solidifies and turns to solid middle lamella. It turns to solid middle lamella. Okay, so that is what we generally see. So now the liquid cell plate turned into solid cell plate. Okay, solid middle lamella. Then that will separate cell into two daughter cells. Now it's separated into two daughter cells. It separates into two daughter cells. Okay, right. Note down that points also. Continue. So it separates into two daughter cells. So this solid middle lamella will separate cell into two daughter cells. Separate cell into two daughter cells. So multinucleate condition arises due to generally, if at all, suppose if it is not divided, if the cell is not divided into two halves, that means there is no formation of phragmoblast, there is no formation of this cell plate, then multinucleate condition arises due to free nuclear division if the cell plate does not occur, then multinucleate condition arises, then multinucleate condition arises due to free nuclear divisions, due to free nuclear divisions. Okay, right, and forms syncytium, This is called syncytium, multinucleate condition or xenocytic. Syncytium or xenocytic, which is nothing but multinucleate. Example, liquid endosperm in coconut. Liquid endosperm in coconut. Okay, right. So this is about the cell plate method in the case of plants. So cell furrow method we discussed, cell plate method we discussed. So this is how the cytokinesis takes place. So karyokinesis we completed, cytokinesis we completed. Okay. So this is what mitosis is. Now, what is the significance of the mitosis? So keep the heading significance of mitosis. So what will be the significance of mitosis? By this, what happened? What we studied in the mitosis? You already know that equal distribution of chromosomes. Is it right? So chromosomes are equally distributed. Equal distribution of chromosomes. Equal distribution of chromosomes. That is one thing. Next, restoration of surface to volume ratio. Restoration of surface to volume ratio. That means whatever the volume which is present in the initial cell, in the two daughter cells also same volume is maintained. So that is called restoration. You don't have a small cells in the daughter, large cell in the parent cell. Everything will be equal. Next, nucleoplasmic ratio. That is nuclear and cytoplasm ratio is constant. Nucleoplasmic ratio. That is nucleus and cytoplasm. Nucleus and cytoplasm ratio is constant. In this, DNA is constant. So there is no variation in the DNA. So the content of the DNA is same. Okay? So it is calculated by the formula NP. Nucleoplasmic ratio is equal to Vn minus Vn by Vc minus Cn. That means where NP stands for nucleoplasmic index, where NP is equal to nucleoplasmic index, nucleoplasmic index. Vn is equal to the nuclear volume, and Vc is equal to cytoplasm volume, that is cell volume. So this is the formula for nucleoplasmic ratio. And next, if you see, the cell repair can be done by mitosis. Cell repair occurs by mitosis in the wounded and damaged. That means wounded and damaged parts are replaced. Wounded and damaged parts are replaced. Example, dead cells of epidermis. If you see examples, dead cells of epidermis, where you can see the replacement. Cells of lining of gut cells, lining of gut cells are also, if they are damaged, they can be replaced. And also RBC. All these are the cell repair mechanisms. Next, it helps in reproduction and grafting in vegetative reproduction. It also helps in reproduction. Mitosis helps in reproduction. Okay. Next, growth in multicellular organisms. Growth in the multicellular organisms. is by mitosis. Growth in the multicellular organisms is by mitosis. Okay, right. So these are all about the significance of mitosis. Significance of the mitosis. So with this we completed mitosis. So now you understood what happens in the mitosis. So why mitosis will occur, how mitosis will take place. I think you understood. There is interphase and M phase. By interphase and M phase, the mitosis takes place. Okay, right. Next is the meiosis. Keep the heading meiosis. Okay. So in the meiosis, what happens? So how does meiosis takes place? Meiosis, why it occurs? All those things. So meiosis. Okay. So what happens in the meiosis? What is meiosis? So meiosis is nothing but reduction division. Shortly, meiosis is reduction division or heterotypic division. Heterotypic division. Why? Because here the cell which is participating in the meiosis will not form the same type of cells. That means one cell participates, one diploid cell, 4 haploid cells are formed. So we call it as a reduction division, that is heterotypic. Heterotypic division. Okay. So why it is called, what is this heterotypic division? Let us see. So where does meiosis occur? Meiosis occurs in sexually reproducing organisms. Meiosis occurs in sexually reproducing organisms. Meiosis occurs in sexually reproducing organisms. So here chromosome number remains constant. Chromosome number remains constant from generation to generation. That is the number of the chromosomes. So whatever the chromosomes which are formed, chromosome number here also remains constant. So only the DNA gets duplicated. Only that is called genetic stability. We call this as a genetic stability. Chromosome number remains constant. What you call it as genetic stability. So this is the chromosome number double during fertilization, which is reduced. That is the DNA which is doubled. The content of the DNA which is doubled, that will be reduced to half by meiosis during the formation of gametes. That means, listen, whatever I told you, in the case of S phase, we see replication of the DNA. So whatever the DNA which gets replicated, then whatever the DNA which gets replicated, so that will form. Simply suppose you see 2N diploid. Where it is taking place? Diploid, always it takes place in the diploid cells. Meiosis occurs in sexually reproducing organisms. That is in which cells? Diploid cells. You can note down. Diploid cells, which occurs in the diploid cells, only in the diploid cells. So diploid cells, if you take a short time explaining, let us see 2N. So now here diploid cells are present. That means what is diploid cell? Two sets of, what is a diploid cell? Two sets of chromosomes. Okay? If you have two sets of chromosomes, in a diploid cell you have two sets of chromosomes. In a haploid cell you have only one set of chromosome. So here two sets of chromosomes are present. So for example, if you take in humans 23 pairs. So these are in sets, two sets. So one is from paternal and maternal. Paternal set and maternal. So 23 from paternal, 23 from maternal. 23 and 23. So the total 46. Here two sets. In two sets they are present. So during meiosis, first it will enter into the interphase. And then what happens? Whatever the sets of chromosomes, just like in mitosis, the number of the DNA gets duplicated. And that will be shared. Here, they form, and in the metaphase, you see they form in cell plate, they will be aligned at the center. So once it is aligned at the center, let us see. Listen carefully. Here, there are chromosomes. So the chromosomes, whatever they are present, those chromosomes are in. Say here I wrote 4. That means here you have 2 pairs are there, for example. So once they enter into the interphase, then what happens? The genetic material gets duplicated. So in the S phase, replication will take place. So whatever genetic material is present, that will get replicated. So now once you enter in this replication of the DNA, that means that you don't see the change in the number of the chromosomes, but its contents get duplicated. What DNA content gets duplicated. Here, in each chromosome, one DNA content is there. Here, in each chromosome, now if you say 2 DNA molecules are present. Here 1 DNA and here 2 DNA molecules in each. But the chromosome number remains same. Now, it enters into M phase. Once it enters into M phase, whatever the content of the DNA which is present here, that will get that. Now, in M phase, you know metaphase. Shortly, what happens? They align at the pairs will align at the line of equator. The pairs will align at the line of equator. That means one is from paternal, one is from maternal. This is a paternal chromosome. This is maternal. So both are same. The size of the chromosomes are one and the same. Paternal and maternal chromosomes. So those are in pairs. They align at the line of equator. And then once they align at the line of equator, Okay, right. So, in the M phase, they align at the line of equator. So now, if you see the chromosomes, what are the chromosomes inside the DNA which is in the phase. Now, the chromosomes will get, whatever the chromosomes are there, they will get separated into, you know, the formation of spindle fibers. So in the formation of spindle fibers, the spindle fibers are attached to this. When spindle fibers attach, now who will separate? Chromosomes will separate. So here the number will get reduced into half. So that is why we call it as reduction division. Suppose here 4 are there, to one pole 2 chromosomes will come, to one pole 2 chromosomes will come. So in the 2 pairs, the chromatids will not separate here. The chromosomes number will become reduced to half. That is what it happens in the meiosis 1. Everything is, what is this? It is called as meiosis 1. So in meiosis 1, see what happened. In meiosis 1, the chromosome number, here 4 are there in the initially. That 4 become 2 and 2 in 2 different nucleus. So finally, by the end of meiosis 1, here you have 2 nucleus. Not nucleus, we call this as dyads. We call these cells as dyads. Dyad 2. So these, whatever it is formed, that is, these will again reduce it to half. Still, this, whatever the chromosomes are present, still it does not form into 4 cells. Now, in meiosis 2, equally we see the formation of, so when the 2 cells are formed, each one will participate into, by meiosis 2, when they participate to the meiosis 2, they undergo mitosis. It is, meiosis 2 is nothing but equational division. Meiosis 2 is nothing but the equational division. So in the equational division, what happens? In the equational division, in meiosis 2, it is just like in mitosis. So here chromatids will separate now, not chromosomes, chromatids will separate. Say suppose, here, in one cell I am showing, again the spindle fibers are formed. When spindle fibers are formed, chromatids will break, just like in mitosis. With kinetochore, they will separate. So what happens? Here already you have 2 DNA molecules in one chromosome. Now here when it separates, then what happens? Each one, they